cold activation of NLRP3. Cold exposure is one of the many known triggers for CAPS. Not direct exposure like an ice cube on the skin, but general exposure. Going outside on a cool day, going into a place with cold air conditioning, exercising, getting sweaty, not getting dry fast enough, and ending up cold. Without treatment, it would be a matter of time before I'd be covered in a rash up my arms and legs, sometimes all over my torso, with fatigue and aching joints. By evening, I'd be a mess, racked with chills and shivers, completely unable to function. Wim Hof I ain't. Treatment with Kineret has been a game changer for me, but even with treatment, cold exposure still exacerbates aches and fatigue and triggers headaches and migraines. CAPS is caused by a mutation in NLRP3. NLRP3 is a gene that codes for a protein, also called NLRP3, that's a cytosolic receptor, meaning that it floats about in the cell, unlike membrane-bound receptors that sit on a cell membrane and sense things outside of a cell. NLRP3 senses certain pathogens and danger or damaged cell signals. And it has many activating triggers, but unlike many other receptors, it's not a matter of this binds to this and then that happens. Most of the activating triggers seem to converge on big cellular problems like potassium efflux, where too much potassium exits the cell, reactive oxygen species, that's a sign of mitochondrial dysfunction, mitochondria being critical for the generation of cellular energy, and damaged lysosomes. Lysosomes contain lots of enzymes to break down broken, excess, or foreign stuff in cells. With all of the known triggers for NLRP3, the specific mechanism of how the activation happens is still unknown. How cold triggers symptoms in CAPS is also a mystery, but there are a couple of ideas about how it might happen. One idea involves proteins that assist with folding. When we make proteins, instructions from our DNA are taken to ribosomes via messenger RNA. I like to think of messenger RNA as a photocopy of DNA, carrying the instructions to ribosomes that are like little 3D printers, making proteins that come out one amino acid at a time like a string of beads. The protein then has to fold into a finished shape so that it can go and perform its function. Sometimes it can take a few tries for a protein to find its final shape, so it needs to safely adjust itself before it can go off and be a finished protein. There are other proteins that assist with this folding process called heat shock proteins or heat shock chaperones. These proteins kind of hold the folding protein back until it finds its right shape. So one idea about the cold is that these heat shock proteins can't bind properly to mutant NLRP3 and have a lower temperature threshold for releasing it. So on cold exposure, mutant NLRP3 gets activated. This was found to be the case for another kind of similar protein, NLRC4, but hasn't been specifically discovered for NLRP3. Like NLRP3, NLRC4 is a cytosolic receptor that can form an inflammasome, and mutations in NLRC4 also cause an autoinflammatory disease that can be triggered by generalized exposure to cold. This paper reported that heat shock cognate protein HSC70 was unable to inhibit a mutant NLRC4 at low temperatures. This worked with one particular mutation, but not two others, reinforcing that the position of the mutation matters. So could something like this also be happening for NLRP3 mutations with a heat shock protein? A couple of papers were published in 2019 that talked about heat shock proteins and NLRP3. HSP70 was shown to inhibit NLRP3 from forming an inflammasome by directly interacting with it. Heat shock treatment increases the expression of HSP70, which will reduce activation of NLRP3. Maybe that's why a hot bath feels so good when I'm flaring. Anyway, they're not sure how HSP is doing this exactly, maybe by changing the shape of NLRP3 or just maybe blocking other proteins from interacting with it so it can't form an inflammasome. Someone's going to work that out soon. I haven't seen any published studies with CAPS mutants yet, but the researchers are proposing heat as a potential treatment. I'd like to see how they think they can make that work. Can't sit in a hot bath forever. So as we covered heat shock proteins, we also need to talk about cold shock proteins. Cold inducible RNA binding protein, CERP, is a member of the cold shock family of proteins, and it gets activated by exposure to mild hypothermia. It also gets activated by UV exposure and hypoxia, which is low oxygen. When it's outside of the cell, extracellular SERP is considered to be a damage signal, which will get the attention of NLRP3. That one might seem obvious, but why cold exposure would send SERP outside of the cell is less obvious. The other thing about SERP is that when it's activated, it's in response to stress. Cold, UV, low oxygen. In stress conditions, our cells need more energy. We make more ATP, and a byproduct of that is more reactive oxygen species, which we know activates NLRP3. Thinking along those lines, a similar idea is that cold exposure promotes other NLRP3 activators. Last year, a paper was published that showed lower temperatures promote monosodium urate to crystallize, which causes gout, driven by NLRP3 activation. Remembering that in CAPS, our mutated NLRP3 is considered to be already in a primed state, it makes sense that our NLRP3 activation threshold is just lower. So any one of these things, or all of them, could be in play. But still, we don't know exactly how and haven't seen any studies on heat shock or cold shock proteins specifically with CAPS mutants. A paper last year described finding elevated levels of cold shock protein SERP in serum samples from patients with adult onset Still's disease. Like CAPS, Still's is a systemic autoinflammatory disease, but it's not well understood, and the researchers discussed NLRP3 activation by the extracellular SERP acting as a damage signal. And Still's disease patients often respond well to the same interleukin-1 inhibitor drugs used for CAPS, but I'm not aware of a cold trigger with Still's, so it could be for them that their inflammation triggers cell death or leakage to get SERP outside of the cell to act as a damage signal, activate NLRP3 in other cells and escalate inflammation that way. It's quite the puzzle, but it will get figured out. A paper came out just last month showing that instead of the primed mutant hypothesis, NLRP3 mutant proteins form cold-sensitive aggregates that did not follow the usual methods of activation. They found that FCAS mutant L353P aggregates were more cold-sensitive than the NOMID mutant D303N aggregates, but the NOMID mutant had a higher number of aggregates, so they speculated it was more constitutively active rather than cold activated, as in the FCAS mutant, which is consistent with what's observed in the different forms of CAPS. While this still doesn't explain how the cold activation works, it's getting closer. It's no secret that cold conditions make a lot of problems for a lot of people, especially people with chronic illnesses. Understanding the activation of NLRP3 inflammation that we get directly in CAPS with general cold exposure might help unlock this mystery for a lot of other people. As always, if we can understand it better, there's a better chance we can develop more effective and targeted treatments.